All right, everyone. Welcome back to Science in the Age of COVID-19. Today, we are super happy to welcome back Angie Rasmussen, a virologist from Colombia. Angie is an excellent scientist in her own right, doing a lot of critical research on this virus and other viruses in her own lab. And she is a force of nature in terms of getting information out about this virus and what's good and what's bad and who's right and who's wrong. She's coming to us. She was on CBS an hour ago, and she has to go right at 3 to be on another media call. So today is our first Ask Me Anything. So please start posting your questions, and we've got some to get started with, and this is going to be really, really fun. Okay, Angie, welcome back.
Thanks, Loren. Really glad to be here as always.
Yeah, this is so fun and so informative. Can you give us a summary of where you think we stand in the vaccine effort?
Yeah, that's kind of the question that's on everybody's mind right now. So there are over 160 candidates currently in the vaccine pipeline, and most of them are at either the preclinical stage or phase 1 stage of development. So those are quite a ways out, but there are, I think, 6 vaccines now that have entered phase 3 clinical trials. And once those are completed, which should be, hopefully as soon as possible. I mean, I suspect that normally phase 3 clinical trials take potentially years because they often look at vaccine durability or how long like the immune protection lasts. In this case, though, I think that they're probably going to issue an emergency use authorization as soon as they have some reliable evidence that the vaccines are effective. So probably a couple months. I would say that I'd guess that maybe a vaccine will have efficacy data to approve one by the end of the year or early 2021. And then it will be several months more to distribute that vaccine within the US. There's still a lot of questions, though, about how that's going to work. If there's more than one vaccine that gets approval, that might speed things up, depending on how well those vaccines work in certain populations of people. There are some vaccines that we know work better in younger people than older people, for example. So a lot of it is going to depend on the data that does come out of these phase 3 clinical trials. But we are on track to have some kind of vaccine that's at least partially protective, probably by the end of the year.
Yeah, can you say a bit more about production and distribution?
Yeah,
Most of the vaccine trials have been going forward with a 2-dose regime, and so for the U.S. we're looking at 600, 700 million doses. How long do you think it'll take us to roll that out?
So this is one of the things, let nobody say that I didn't, give praise where it was due. This is one of the good things about Operation Warp Speed, is that it has really offset the financial risk to the manufacturers by basically making them able to manufacture these vaccines before they're approved. So normally companies would not do that. They would not invest millions of dollars in making a vaccine that they don't know that they're ever going to be able to sell. But in this case, Operation Warp Speed has basically paid those costs. So they are manufacturing them now, but that's a very good point, because if you're manufacturing 100 million doses of vaccine, but it's a prime boost regimen, meaning you need 2 shots to get full immune protection, that means that you're actually only making 50 million effective vaccine doses, because each dose you have to get 2 doses, or each person has to get 2 doses. So that is going to be a big factor, and that will really depend a lot on whether, say, a viral vectored vaccine is approved, which might only need 1 shot, versus an mRNA vaccine, which probably needs 2. So that's a great question. With regards to distribution, that's going to be something that I think is going to be very controversial, because the vaccine is going to be made available based on priorities, and it's not entirely clear to me how those priorities are determined. I think everybody agrees that, you know, frontline healthcare workers should be a prioritized group, but otherwise, like, are people going to prioritize vulnerable people who are at higher risk because they're older, they have pre-existing medical conditions? Are they going to prioritize other essential workers who are not healthcare workers, who might be at higher exposure risk? And how are they going to ensure that access is equitable across many of the groups? As we have seen, the pandemic has exposed some real disparities within our society. So how is the distribution of the vaccine going to address that and make sure that people have equitable access? These are all really important questions, and it's not entirely clear to me how those things are going to be decided.
Yeah, we'll definitely have to make it up as we go along.
Yeah.
I'll just ask you one more question about vaccines and then turn it over to Sarada. You mentioned the pro of Operation Warp Speed paying for these doses. Can you say a bit about the downside of, let's say, Moderna producing 250 million doses, and then when we get the results from phase 3 back, it either looks like not great or okay, but we can do a lot better? What happens then?
So I think that really does depend on how okay it is. You know, I have a lot of questions specifically about the Moderna vaccine in terms of its reactogenicity. In the phase 1 trial, there were 4 people that withdrew because of severe adverse effects, and that's about 10% of their trial participants. So that, I mean, if that is also the case in a 30,000 subject phase 3 trial, I mean, that doesn't sound like actually a very acceptable safety profile to me. So that's going to be one thing. I think that, you know, the downside is obviously just a waste of money. I think that if the vaccine doesn't meet the standards for approval, either for safety or efficacy, then, you know, we wasted millions of dollars. But I'd rather waste money than wait, so that, you know, delaying the point at which people could access the vaccine.
Yeah.
So I think one of the other downsides to Operation Warp Speed is the stupid name. I think that it really has put a lot of questions in people's minds about whether the process is being accelerated to the point that they're cutting a lot of corners. And right now, I mean, at least in the short term, the phase 3 trials are set up to determine short-term efficacy and short-term safety. We really can't look at long-term efficacy or durability or long-term potential safety issues, because we just are going to approve the vaccine before that. So that is one shortcoming of this accelerated process. But I mean, I would feel comfortable taking a vaccine that has been shown in a phase 3 clinical trial, even on this shortened timetable, as long as it's shown to be protective. And the number that I've heard is 50% protective, meaning that 50% of people who get the vaccine will be protected from severe COVID. Now, it's totally possible that the vaccine's not going to be great in terms of efficacy. Like 50%, like that's half the people who get the vaccine are still going to be potentially susceptible. I mean, so far, the preclinical non-human primate challenge data that has been generated does look like the vaccine is protective in the majority of animals against severe disease. Many of these vaccines do not produce sterilizing immunity, so there will be questions about, even if people are protected from disease, which is a huge public health benefit, will they still be able to transmit the virus? I think that's a huge question, especially since people won't all be getting immediate access to the vaccine. So I think, you know, some vaccine that's somewhat effective is better than no vaccine at all. But I think that, you know, we still, once there is a vaccine that's approved, it's not going to be the end of the story for vaccines. Vaccine development will continue. We'll need to keep monitoring people who've gotten the vaccine to look at durability and long-term potential safety issues, or potentially rare, extremely rare side effects that wouldn't be apparent until you actually vaccinate a lot of people and have enough so that those rare, severe adverse effects would emerge. So there's still going to be a lot to do once it's done, even if we get to approval at warp speed. It's not like we're going to be, okay, let's close up shop and we're done here.
Yeah. So let me just add on an audience question before I turn it over. How is this whole high risk group thing? What are the logistics of that? Is it a doctor's note, or do you just present yourself as high risk? Or, I mean, again, how do you think that plays out?
You mean for prioritizing people who?
For vaccine or therapeutic or, yeah.
Yeah, that's an excellent question. I don't know how you would be asked to prove that. I mean, presumably if you have diabetes, if you have a heart condition, something like that, you would have a diagnosis to demonstrate that. Obviously, if it's just going by age, then you could just show your driver's license or something like that. Although it is a good point to bring up that not everybody has ID that has their birth date on it either. But that's a really good question that I don't completely know the answer to. I'd pretty much just be guessing, because I don't really know how you would be asked to prove that. And I don't know the answer to that is the short answer.
Yeah. Okay, Sarada, you want to talk about therapeutics?
Thanks, Angie. So on a related note, here's a audience question on therapeutics. What are the therapeutics that are working the best? Could you list these by age group? I mean, could you just in general summarize the current therapeutic landscape for us?
Yeah, so there are a number of therapeutics that are in clinical trials. Some of these are immunomodulatory drugs that are designed to reduce inflammation or specific aspects of inflammation. In terms of therapeutics that are already available for use, there's remdesivir, which is an antiviral. It blocks the enzyme that copies the viral genome. It has some effect, but in the patients that it has been tested on so far, the main benefit to getting remdesivir is that it reduces the time that a patient is hospitalized for. So it doesn't actually provide a significant mortality benefit in that it saves lives. It just reduces the amount of time that somebody is sick, which is still, again, a big public health benefit, especially when one of the biggest problems that we're facing are overwhelmed healthcare systems. So getting people out of the hospital faster is a good thing, but it's not ideal in that it can't cure people and save the life of somebody who would otherwise have died. So there may be a benefit for remdesivir if it's given earlier. There are some issues with that because it's not a pill. You have to have an intravenous infusion of remdesivir. So they aren't giving it to somebody who just tests positive for coronavirus and has no symptoms. You have to actually get sick before getting it. Mm-hmm. So it may be that remdesivir might have some use in the future if there's enough of it, or they figure out a way to treat people with it very early on when your viral levels are still increasing and the virus is replicating. That's what it blocks. But by the time people are sick, what's causing a lot of the disease is actually the host's response to the viral infection. So if somebody's in a hospital on a ventilator, just reducing the amount of virus that they have circulating in them is not going to necessarily do much to treat their actual disease. For those patients who are severely ill, they've found that dexamethasone, which is a corticosteroid, it's very cheap, it's widely available, it's an immunosuppressant that blocks inflammatory responses, does have a benefit for those very severely ill patients. But that's sort of the opposite, because you don't want to give dexamethasone to somebody who has just gotten sick, because it is an immune suppressant. So it will suppress the immune responses that the body would normally use to control the infection. And for people who end up having very mild or asymptomatic disease, it's likely because they are having these effective immune responses that are clearing the virus before it can replicate to such high levels that they're getting this cytokine storm, this inflammatory disease from it. So those are good. It's good to have any kind of therapeutic in the toolbox, but we definitely need therapeutics that are more effective at treating severely ill people, either by a combination of reducing the amount of virus that they have circulating, or by reversing their pathology through modulating their host responses. And so I think that's what a lot of the different therapies that are in clinical trials right now are doing, one or the other of those things.
So on a related note, what's your take on monoclonal antibodies? Do you think monoclonal antibodies would make a difference in this fight, or what's your take?
Yeah, so monoclonal antibodies are interesting because they can kind of do both things. So they can be antiviral, but they can also have immunomodulatory effects. And we've seen this with Ebola, actually. So there is a consortium run at the La Jolla Institute of Immunology, led by Erik Snapp, where they've tested huge panels of different therapeutic monoclonal antibodies against Ebola virus. And what they found was that some of the neutralizing antibodies are in fact protective in mouse and non-human primate models of infection and disease, but also they had some non-neutralizing antibodies that were also protective. And this is probably because those antibodies will bind the virus and be taken up in antigen presenting cells, or any cell with an FC receptor that can bind the non-specific end of the antibody. And it takes up the virus and allows those cells to respond with other immunomodulatory effects that can be protective in an infected host. So I think that monoclonal antibodies have a lot of potential. So far, I'm not convinced that just using convalescent plasma is very effective. I know there are a number of trials going on for that too. So it may be, but I think monoclonal antibodies have a lot of promise just because you can more directly target specific parts of the virus, and you can select for antibodies that are doing very specific things, both in terms of their antiviral effects as well as their potential immunomodulatory capabilities. So I think that, that certainly there is a place for monoclonal antibodies in the therapeutic landscape, and I know that some are undergoing clinical trials right now.
All right, thanks. Loren, did you?
Yeah, Angie, I wanted to follow up. There's one therapeutic that you didn't mention. What do you think about early interferon treatment?
Oh, early interferon. I think that also has a lot of potential. So some of the host response data that has come out does suggest that infection period, regardless of disease severity, is associated with suppression of type 1 and type 3 interferon responses. And interferon, is an antiviral cytokine. And that's actually how it was named. It was discovered to interfere with viral replication, so they named it interferon. I think that, it's certainly something that should be evaluated along with everything else. Now, I think if you have acute COVID, you're probably not worried as much about the side effects from interferon, which are just like not fun. I did my postdoc working on hepatitis C virus, and back then the only treatment that was available was interferon and ribavirin, and you had to be on interferon for 18 months. Usually it didn't work very well. Hepatitis C virus is very different. It's a chronic disease and a chronic infection. And interferon is associated with suicidal depression and chronic flu-like symptoms. So 18 months of that was very difficult to justify for a treatment that didn't work very well. I think, though, a short course of interferon to treat an acute SARS coronavirus 2 infection, those side effects would probably be acceptable if it does in fact work, and it's effective at treating those. It's quite expensive, though, for pegylated interferon, which is more bioavailable. So that's one thing to consider, but I do think that those trials are in progress now.
Are there biosimilars for pegylated interferon? Do you know?
That's a good question, and I don't know. There may be. I haven't been keeping up as much with the therapeutic interferon field since I stopped working on hepatitis C virus, but There may be, because certainly, I mean, interferon has potential for treating many different viral infections. And especially if you're going to use it to treat anything chronic, or where you need to be on it for a long time, it would be desirable to have biosimilars that had a more favorable profile for the people taking it.
Yeah, I mean, my naive read of the reports coming out was that it was short courses that people were trying. Yeah, I hope that ends up being the case.
Yeah, me too.
I didn't know about those brutal side effect profiles.
Yeah, I mean, so for genotype 1 HCV, which is most common in the US, it only has about a 50% efficacy rate at treating that. And so a lot of clinicians, I mean, this was back in like 2009, 2010, before the next generation protease and viral protein inhibitors came out, that now are much more effective and have a much more favorable safety profile. Many clinicians would just not treat their patients with HCV unless they showed signs of progressive liver damage, because the treatment itself was such a reduction to their quality of life.
Yeah.
And ribavirin can cause hemolytic anemia, so that was also part of the regimen. So yeah, 18 months of depression, flu-like symptoms, and hemolytic anemia was, you know, understandably, makes life kind of suck for those HCV patients.
Yeah. Hey, I want to change gears a little bit. So testing sucks in the US. It has always sucked. Now it seems to be really bad. My friends got tested, and most of them had 2-week plus turnaround times. How do we fix this?
This is a really good question, and it's a huge problem. I mean, part of the answer is just continuing to increase testing capacity, and some of the issues have to do with the fact that the testing providers are either using types of tests where there are significant bottlenecks in processing them. They're not always done with, you know, high throughput automated testing systems, and that makes a big difference in terms of throughput and turnaround time. So that's one problem. But the other issue is that we just need more different types of testing technology. And there has been a pretty vocal movement of people led by Michael Mina at Harvard who have advocated for approving like do-it-yourself at-home tests that would be very rapid. And the reason why the FDA hasn't approved these tests is because they're not as sensitive as the PCR test. But what Dr. Mina has advocated for, and I agree with this, is that maybe you don't need extreme sensitivity to tell if you are potentially infected and capable of transmitting the virus to others, because that's the real question. Like, if you have really low levels of RNA, viral RNA present that are right at the limit of detection, you're probably not shedding so much virus that you would be able to infect another person. So it's important for you to know that you're infected so that you can isolate yourself, but it's also, and potentially seek medical care, but it's not necessarily going to make a big difference in terms of your ability to spread the virus around. If you have these daily, you know, tests that could be done, you know, once a day or a couple times a week, that would be very cheap and easy for everybody to get, then, you know, maybe you could find, even if they weren't perfect in terms of their sensitivity, maybe you could identify people who were more likely to be spreading the virus and more likely to be contagious. So I think it is really important that this issue get worked out. And, you know, I think the FDA has been somewhat reactionary in how they've gone about issuing emergency use authorizations for different types of tests. So initially, there was a whole crisis back in the spring where the CDC was the only one allowed to make a test, and then the test didn't work, and then people just said, you know, screw it, like, we need a test. So they started making their own tests, and so then the FDA started approving those tests. And then when serology testing was developed, they kind of did the opposite, in which they reacted to the criticism that they weren't approving tests quickly enough for the molecular diagnostics. So they like approved everything for the serology tests. And the result was a bunch of antibody tests that basically were no more sensitive and specific than like phenycoin. So those tests are pretty much worthless. So now they've kind of gone back and they're like, well, these tests aren't as sensitive as the PCR tests, so we're going to hesitate about approving them. But I think they really do need to start getting some additional testing on the market. We also need to do things like we need to have better surveillance so that we know where to redirect testing resources to make sure that communities that are in the most need of testing and have the most testing demand are able to meet that demand. And we can anticipate that by doing things like wastewater sampling and seeing if virus levels in the wastewater are going up. That might indicate that there is a number of cases that are about to be found in a given community, and you could potentially redirect testing resources to those areas. But you're right, a 2-week turnaround time is completely unacceptable for a coronavirus test, because not everybody is going to be able, without a test, to isolate themselves or to miss work. They may not have sick leave. There are a lot of reasons why that just is really bad for people who don't know whether they're coronavirus positive or not. And I think that it's really something to consider. Another alarming thing is that testing overall has been decreasing. So we were at peak testing at the end of July, but now the actual number of tests performed has started to go down. And it's not completely clear to me why. The COVID-19 tracking project has been tweeting about this over the last couple of days, but there has been a reduction in testing overall, which is also not good. And it certainly only contributes to this problem of bad turnaround times for testing.
Yeah, I mean, so do you think there's anything that we could do at the sociological level with testing? So can we get companies to, for instance, acknowledge that if it's 12 days after you've gotten the sample, that, you know, maybe you should just throw that out and reply to the person, look, I'm sorry, here's your money back, and we're going to prioritize some tests that just came in this morning. You know, I think it's going to be hard to get companies to go along with that, but it would make the same number of tests be much more useful.
Yeah, I mean, I think it's worth a try, but I think you're right that it's going to be very difficult to get companies to do that, because if you ask like Quest or LabCorp, they're going to say like, oh, our testing is great. Like, we are doing such a great job and we're better than we were like back in March, which is not exactly a huge achievement. I mean, one would hope, but a lot of times, you know, and even when the White House coronavirus task force like actually talks about this kind of stuff, they just keep making the point that we're doing more tests, but we're still not doing enough. And there's not a lot of accountability for why the turnaround time is so bad. And sometimes I think people don't even know who's actually doing the test. You know, they might not know where their doctor's office or where a given hospital is going to actually send their sample to be tested. So I think, you know, that's, in a perfect world, that might be one solution, but I think that's pretty unlikely, just knowing the way that, you know, these testing companies work, especially the larger ones that have kind of monopolized the testing market for everything, like Quest Diagnostics and LabCorp.
Yep. Okay. Sarada, you're back up.
Yeah, we have an audience question here. Brian, please unmute yourself and ask your question.
I just wanted to know what would be the sort of minimal turnaround time for testing for it to be effective public health? Is it like 2 days or?
Probably at the most 2 days. People should get a test back within 24 hours, ideally, because, you know, really, I think if you think that you've been exposed to coronavirus, like, you should isolate yourself, or quarantine yourself would be the correct term, until you have a positive test. But if you have a positive test, you want to know as soon as possible, because if you're negative, you think you might have been exposed, that might change the amount of time that you actually have to quarantine yourself. It also might suggest that maybe, you should go get another test like within 2 weeks, but you might not have to necessarily take off work. So as soon as possible is really my answer, but I would say within at least 2 days.
All right.
I mean, I guess the other thing I should just make one more point is that if we are ever able to get community transmission back down to a low level, the way that we will be able to keep it that way is to have rapid testing turnaround so that contact tracers can go out and talk to the people that you might have exposed if you're the one testing positive. So the sooner that contact tracers can go out and do that, the more effective that method of containment is going to be. So again, testing turnaround time needs to be very, very short, probably at most 2 days.
Brian, do you have a follow-up question?
No, thank you. That makes a lot of sense. Thank you.
Thank you, Brian.
You
Thank
bet.
you. Our next audience question is from Erik Snapp. Erik, could you unmute yourself and ask your question? Erik?
Yeah, can you hear me now?
Yes.
So I had a question. So the way that we think about coronavirus infection, at least the way it's been presented in public health, is that it's sort of binary process that you either have enough virus to get infected or you don't have enough exposure. But we also hear about people that don't fit into the traditional comorbidities, like young doctors getting sick and dying from coronavirus. And so is it that you could also get a higher dose of virus leading to more severe illness? I haven't heard any real discussion of that.
Yeah, that's an excellent question. And the short answer is we don't know. But the less short answer, I guess, is probably that's true. With many viruses, there is definitely a relationship between infectious dose and disease severity, but not always. And there's something that's important to consider whenever you're talking, especially about RNA viruses like coronaviruses, and that is that not every single viral particle that is produced by an infected cell is actually infectious. And when you have, and so what we call this the particle to PFU ratio. So PFU is a plaque-forming unit that's basically a single infectious virus. And it's measured by its ability to form a plaque or to basically kill cells on a culture plate. That's how we quantify infectious virus. So if the particle to PFU ratio is high, meaning that there's a lot of particles, but only a few of them are infectious, that can actually at high doses have a sort of counterintuitive effect where it reduces severity of disease, probably because those particles can either be what are called defective interfering particles, meaning that they might infect a cell, but they don't complete the replication cycle, and that actually can have a negative effect on the number of infectious viruses that are produced by that infected cell versus another infected cell that's infected with an actual competent infectious virus. They can also, by exposure to those DI particles, it can also activate antiviral immune pathways in cells that might be infected and essentially create an antiviral state that is better at preventing infection of the actual infectious viruses. So for example, some of our lab stocks of Ebola have a high particle to PFU ratio. And if you infect mice with a very high dose of the virus based on PFUs, you're giving them so many of these non-infectious particles that actually fewer mice end up dying. So there probably is a relationship between dose and disease severity, but we don't really know like at what particle to PFU ratio or what ceiling, if there is one, that this virus will hit where it might not actually make any sort of difference. We also really, dose is not the only thing that determines disease severity. So some of my work is actually about the effect of the host response. And we found in a genetically diverse mouse model of Ebola virus infection that you can infect all of them with the same dose And some mice will die. Some mice will die of what looks like Ebola virus disease. So they have hemorrhagic disease and many other features of human Ebola virus disease. And some of the animals like lose a little bit of weight and then completely recover. So a big part of it is dose, but a big part of it is also how an individual responds. And that's probably based on a combination of genetics, epigenetics, their environment, their general health status, things like diet, behavior, stuff like that. So there's really a lot of factors going into this, and it's not a straightforward answer. But back to my short answer, nobody has yet shown a correlation between dose and disease severity. And we actually don't know what the minimum infectious dose is for people in the real world.
Great, thank you.
You bet.
Thanks, Angie. Thanks, Erik. So, Angie, on a related note, what's the current understanding of long COVID, especially those with mild to no symptoms?
Yeah, that's a great question. And I suspect, like with many things, my answer is we don't understand that much about it. Right now, there has not really been a sort of definitive study kind of describing long COVID and all the different forms it can take and quantifying how frequently it occurs. But there are a number of anecdotal reports that are ever growing. I mean, it really does seem like it's a thing. And it's not particularly surprising to me, at least with regards to the chronic lung injury that some people have reported. So we already know that SARS Classic and MERS coronavirus can cause fibrosis in the lung, which is basically scar tissue. And that's really irreversible lung damage. So it's not shocking to me that this related beta coronavirus is also causing that type of response in some people. Some of the other long COVID symptoms are a lot more perplexing. You know, there are these neurological sequelae that are starting to be reported, and it's not completely clear if those are due to sort of residual damage that was done by the primary infection, the way the lung damages, or if it's because of some kind of persistence of the virus itself in an immunologically protected niche. And that has been observed for other viruses as well. With Ebola, a number of Ebola survivors have reported all kinds of different long-lasting neurological effects, including insomnia. Some people have had problems with their vision. Some people have had issues with their mental health, irritability, depression, all resulting from their recovery from Ebola. And it's really hard to try to figure out the mechanisms of many of these things, because you can't, you know, you're not going to do a brain biopsy on somebody who has insomnia, probably. And in a lot of cases, you just really don't know what's actually causing this. For Ebola specifically, I mean, there's a lot of stigma associated culturally with people who are Ebola survivors. So if somebody's depressed after recovering from Ebola, is it because they're now a part of a marginalized population? Or is it because they actually have some physical neurological damage that was done by the infection? And it's really difficult to try to figure those things out. There have been these reports of long-lasting neurological sequelae for COVID-19 survivors, as well as problems with the heart. I've heard of long-lasting renal problems. I've heard of long-lasting sort of blood clotting issues. And that's also something that happens with other infections. So people who have severe influenza are more likely to have myocardial infarction or a stroke after they recover. So there's really a lot we don't understand about acute viral infections in general and the long-term consequences of having them, but it's becoming more and more clear that we are going to need to take a very close look at this with COVID, because some of these reported sequelae are so severe that, you know, they may be permanent, and that may lead to an increase in the number of people who are disabled, people who need some type of long-term medical care because of these chronic conditions that have developed as a result of them having COVID. So this is definitely an area where we don't know that much right now, but we need to start focusing on this, because as you pointed out in your original question, some of the people who are reporting having these long-term symptoms did not have severe COVID-19. They weren't hospitalized and they had relatively mild illness. So can you really call something mild if it's impairing you over a long-term period of time? I mean, I don't personally think so. I think that if a mild acute viral infection results in a chronic condition, that's actually very serious, and it has the potential to be a long-lasting public health problem that's with us for a long time.
Exactly. So this makes me wonder what exactly causes the sequelae. I know you touched on this, because is the virus itself infecting these tissues, or is it the aftermath of an immune response as a result of this infection? But then if it's a mild infection, do we anticipate the immune response to be so strong that you have these long-lasting damage to these other tissues, like the brain fog, the chronic fatigue?
Right. So I mean, that is a really important question. And I think the answer is that it doesn't necessarily have to be like a cytokine storm type immune response to nonetheless create sort of this long lasting pathology. So we already know that there's a number of viral infections that can result in this that are not necessarily associated with this widespread inflammation. So there's post-polio virus syndrome, which is a sequelae that can sometimes emerge years after a person has recovered from poliovirus. And, you know, polio is, I mean, it's associated with paralysis, but that's because the virus itself kills motor neurons. It's not because there's a huge systemic inflammatory reaction. So we just really need to know a lot more about how maybe, you know, not that, I mean, clearly for the lung injuries, the virus itself is doing damage to the tissue, and the immune response to the virus in those tissues are causing this fibrosis to form. For some of these other things, though, is it more like even a mild infection can sort of recalibrate the immune system? I mean, this has been proposed for a mechanism for how chronic fatigue syndrome or myalgic encephalomyopathy, I think that's it. MECFS, chronic fatigue syndrome. That's been proposed as a mechanism for how that occurs, that basically a viral infection or some other type of initiating basically changes the way the immune system fundamentally works or is responding to things in those patients. So we really just need to do a lot of research to try to figure out which of those mechanisms are sort of at play here. And it may be that it's a combination of all of them, that it's damage from the virus, potentially viral persistence, or that it's somehow like a recalibration of the immune or inflammatory response that's causing this.
Right. So on a related note, since we touched on syndromes, could you speak to the correlation, if at all any, between Kawasaki syndrome and the SARS-CoV-2 infection?
Yeah, so they're now not calling it Kawasaki syndrome for the COVID stuff. It's, I think, now called MISC, or multi-inflammatory systemic from COVID or something, or syndrome from COVID, MISC. Okay. But it's basically a aberrant inflammation that affects multiple organ systems. And it's rare, but we don't really know anything about like what puts a child at risk of getting that versus somebody, you know, a child that has a mild illness and recovers quickly. To my knowledge anyways, like nothing really is known about what places somebody at risk for developing that or not.
Right. Clearly more research needs to be done, I guess, to address
yeah, that's the general theme of every Q&A I do. It's like, I don't know, we need to do more research.
Fair enough. All right, so we now switch to an audience question. Nelson, please go ahead, unmute yourself and ask your question.
Thank you. My question is just whether you can give us a explanation, sort of mechanistic explanation of how these new RNA vaccines actually are intended to work.
Sure, yeah, that's a great question. So the main thing about these RNA vaccines is that they are intended to simulate messenger RNA, which is normally how our cells express genes. So your genes are encoded in your DNA. That is transcribed into messenger RNA, which gets exported from the nucleus and goes to the ribosomes, where it's translated into protein. The mRNA vaccines, like the Moderna vaccine, are basically pieces of messenger RNA that are encoding the spike protein, which is the main antigen on the surface of the SARS coronavirus 2 virus particle. And that's what you make antibodies against. So basically, it's giving messenger RNA, encoding spike to your cells, and having your cells make the protein for your immune system to then respond to. Now that mRNA has to get into the cell, so the RNA vaccines are packaged with a proprietary lipid that allows them to be taken up by the cells so that they can get inside the cells into the cytoplasm, meet up with a ribosome and make that spike protein, which then is recognized by the immune system as foreign. This is not the most immunogenic way to make a vaccine. Usually, that's why live attenuated vaccines or viral vectored vaccines tend to be more effective, because they actually have a replicating virus that will sort of ring more alarms for your cells and trigger a more robust immune response. So that's probably why we need 2 shots for the mRNA vaccines, because you just need a little bit of extra stimulation to have them be as immunogenic as you want them to be to produce long-lasting protective immunity.
I just had a quick follow-up to Nelson's question. Is it known what cell types are taking up the lipid droplets, and if that's at all relevant to production of the antigen?
That I don't know. I would imagine, though, that it's probably muscle cells because it is an intramuscular vaccine. But that's a great question that I don't actually know the answer to, and I don't know if anybody's actually studied that.
So that's similar to what a follow-up question I wanted to ask, and I was going to ask about the route of administration. So if it's an IM injection, then the mRNA just gets into cells locally near the injection, and those are the only cells that will produce the spike protein. It seems like there'll be a small amount of spike protein.
Yeah, and that is a great point. That's probably part of the reason why they need a second shot. It's also one of the reasons why some people have started to make these next generation RNA vaccines that are actually RNA replicons. So they are an incomplete viral genome for the self, but it doesn't actually make any virus. And in those vaccines, I know one is being developed at the University of Washington, and there are a couple other companies that have candidates like that. So basically, in that case, the RNA itself is replicating rather than the mRNA vaccine, which just eventually gets degraded. So that they maintain that mRNA for longer so they can express more protein. And that is thought to be a disadvantage of the mRNA vaccines, is that they aren't generally an unstable molecule and it can be degraded quickly. So these self-replicating RNA vaccines sort of solve some of that problem by keeping the RNA around to sort of maximize spike protein production. But it's a great point, and it's well taken that there are limitations to how much spike protein can be produced, and that probably does impact the robustness of the immune response.
Thank you.
You bet.
Thanks, Angie. Our next audience question, Nathan, please unmute yourself and go ahead.
Okay. Yeah, what is the likelihood that COVID-19 recurs seasonally, like the flu?
Well, I think pretty low, just because we've seen this summer that this isn't really seasonal. I think that that was a big question for me in the spring. And I know that a lot of people sort of made assumptions that it was going to be seasonal, like the common cold coronaviruses or influenza. There's a lot more to seasonality, though, than just the weather outside and just the season. A lot of it sometimes has to do with the wildlife reservoirs of virus, as well as its circulation in the southern hemisphere, places where people are being exposed to zoonotic sources of the virus. So with influenza, not only does it infect wildlife, but it also circulates in domestic animals. So chickens, ducks, pigs, and that's oftentimes how people are exposed to new flu strains. So some of that goes along with like those animals' breeding seasons, and when people are going to be exposed to those types of animals, and that can have a seasonality component to it as well. So seasonality is like more complicated than just, you know, is it spring, summer, winter, fall? But it also is important for viruses like flu that are constantly emerging into the human population from an animal reservoir. With this virus, it already emerged from that reservoir and it's not constantly re-emerging from it. There was only the initial entry into the human population, and now it's being transmitted from person to person efficiently. So it's less likely to have a seasonal component, other than the types of behavior that would lead people to be at higher risk of contracting it. So for example, in the winter, people are more likely to be inside in close quarters than they are in the summer. In some places that are very hot in the summer, people are also more likely to be inside because it's air conditioned. So it really, that can be sort of seasonal, but this virus doesn't appear to be seasonal like the flu.
Angie, I had a related question. So, you know, given that fall and winter are fast approaching, what do we know yet about viral exclusion with COVID? Because we're going to have, you know, flu, the RSV, rhinoviruses, picornaviruses. But what's your intuition about, are these going to exclude each other, or are they going to be collaborate with each other? Do we have any idea?
I don't know that they're gonna collaborate necessarily, but I don't think that there's any evidence that they'll exclude each other either. And we do know that at least there were several reports of people in China at the beginning of the pandemic who were co-infected with influenza and SARS coronavirus 2. I don't know if that is going to have any sort of synergistic effect on making disease worse, or potentially like sort of canceling each other out, making disease less bad. That's, you know, I think that's really uncharted territory. I think the biggest risk for me is I'm less concerned about, co-infections and like super influenza coronavirus like disease, as I am with just like the strain that this will put on healthcare systems. If people are getting flu or coronavirus or RSV, or, you know, having an asthma attack because they got rhinovirus, and they have to go to the hospital, all of that is going to place a higher burden on the hospital system because they're also dealing with tons of coronavirus infections. So I think that the biggest risk is really the strain that it will have on the healthcare system, because so many more people will be competing against each other effectively for the same finite resources to treat severe respiratory diseases.
Yep. Okay, Sarada, you back up?
Yes. So here's a question from Dr. Adam Hantman here in Janelia, and I'll probably join him in asking this question. What, according to you, is the next 6 months going to look like? Two versions, ideal and predicted.
Okay, so ideal is that we shut down for 6 to 8 weeks to reduce community transmission. During that time, we increased our testing and contact tracing capacity, and then we can slowly reopen and sort of reemerge. During that ideal shutdown, there would be increased coronavirus stimulus packages passed to allow people to be economically supported through a stay-home order like that. But I mean, granted, I don't think staying home for 6 to 8 weeks is ideal, but I think that is what is necessary to get back down to where we need to be in order to start safely reopening. Presumably, I mean, shortly thereafter, a vaccine will be available, and then we can start reopening while we are beginning to ramp up vaccination efforts, and that will be that will sort of lead to, at the end of that 6 months, sort of the beginning of the end of the pandemic. What I think is actually going to happen is that we're not going to shut down. In some of these places, community transmission will remain high because we're not doing enough testing. We have too much transmission occurring in the communities to allow for contact tracing. And some places are still making decisions based on political considerations rather than good public health guidance. So that sheriff in Florida, for example, who forbid people from wearing masks at all at the police station. When people are doing stuff like that, that basically means they're going to have more transmission in their communities. If people continue to believe misinformation that downplays the severity of the pandemic, they're probably going to engage in riskier behavior. It's going to put them at higher exposure risk. So I think that we are going to be grappling with high levels of community transmission for some time. And we might start to see transmission increase in even states like New York, where transmission has been kept very low, as people return from around the country to their colleges and universities and bring in new cases of coronavirus from out of state. I think even in states where we're starting to see case numbers go down, we have the potential to see them go back up if we continue to make sort of the same mistakes that we did with reopening the first time around. So I think we are going to be sort of grappling with a patchwork of rising and falling case numbers around the country until we get a vaccine and until we get enough people to take that vaccine that there is effectively herd immunity to reduce that transmission. But I think my outlook is pretty pessimistic. I think that there is also the potential for a real crisis as flu season starts if people don't get flu shots, especially. And I know that, you know, the sort of anti-vaccine movement has really co-opted a lot of their language and their branding, for lack of a better term, and applied it to the pandemic, further reducing trust in public health guidance. And overall, I mean, I think that we have the potential to be in a very serious situation in the fall when school opens back up around the same time that flu season starts, when we still don't have a vaccine. So sorry to end on kind of a Debbie Downer note, but I'm not super optimistic about the next 6 months.
Wow. Okay.
Yeah, way to end.
Yep. Thanks, Angie.
I know. You're like, never come back.
Wait, would you like me to present another question so that we end on a better note? No,
Sure.
Just kidding. I know you probably have a hard stop.
Yeah, I have a hard stop in 2 minutes.
All right. I guess we can never have enough of your time. We have many questions, but I guess we have to say a huge thank you to you and let you go, Loren.
Yeah. You go save the world, Angie.
Thank you. And it's actually not a media opportunity. It's a call with a collaborator. But
that's much better.
That's much better, and especially with regard to, I don't know, saving anything, but at least doing science. But it's really been a pleasure to be back here with you guys, and I'm happy to come back anytime for something similar.
We'll take you up on that, Angie. This is so awesome.
Well, thank you so much, Loren and Sarada. It's always great to talk with you guys.
Thank you.
Yep. All right.
Stay
Stay safe.
Care.
Stay sane.
You too. Everybody stay safe. Bye-bye.